Wonder where the laser pointer is. Anyways, I'm going to be talking today about this difference in delimiting species using, say, reproductive isolation versus detecting coalescent lineages using coalescent models to determine independent evolutionary lineages. To do this, we use this empirical data set, which some of you might recognize. This is the coin and ore data from 89 and 97. They're fruit fly data. Using some updated measures on reproductive isolation from Jokovic 2012. And essentially, their theoretical predictions from this empirical data set was that as reproductive isolation increases with time, so does the genetic distance between taxa. They also put very clear demarcations on how much difference was needed. So for reproductive isolation, they put this measure here in black, 0.93 according to their index, 903. And then they separate genetic distance dependent on overlap in breeding range. So sympatric species needed very little genetic distance under their model, difference in genetic distance under their model, assuming that reinforcement was deterring heterospecific matings. They put forth a demarcation on allopatric species that required a greater genetic distance. This is due to the fact that there's probably not heterospecifics around, so you don't need something inhibiting mating at low genetic distances, such as reinforcement, as they said in sympatry. So that gave us some very clear demarcations in the sand of what they said was a good species and what was not a good species according to their model. And then we could use alternative models, different models, such as the multi-species coalescent, to test for concordance between what they say is a good species and what, say, another model says is a good species. So we're going to use this figure from Halid and Drummond 2010 to just kind of describe the MSC process real quickly. So coin and ore's process is essentially the biological species concept. They're looking for reproductive isolation. The multi-species coalescent, on the other hand, looks for independently evolving lineages. And we've seen this shift in systematics over the last decade or so, and cited mostly by DeKiros' work in the late 90s, early 2000s of this lineage, a species of independent evolutionary lineage. And in that same vein, more recently, we've had the MSC, multi-species coalescent, provide a way to identify independent coalescent lineages based on sorting of alleles. So, very briefly, the MSC model, everything has a distinct population size. This is a prior that you need to specify in the model. Additionally, there's a start and stop time for each lineage. So, for example, species A and species B stop here and become species AB. Likewise, if you go further back in time, species AB coalesce into species ABC, the ancestral species of the whole group. Some prior parameters that need to be assumptions that are included in the model is that speciation is instantaneous, which is to say after C has split off, there's no gene flow back into A and B. And another assumption, as well as that perhaps alleles are partitioned into some number of lineages. Perhaps it's better that A and B go away as independent things, and rather it's one species, AB. And that's the whole goal of species delimitation, right, is saying, are these things better as 2 or 1 or 3 or 5 or whatever? So that's the beauty of the model, is it's able to tell you with what probability that you have more than one lineage. So what were our expectations? We expected that as posterior probability of being independent lineages increased, we would see a similar increase in genetic distance, total isolation, these measures that coin and ore use to delimit species. We would see the multi-species coalescent also showing that as those indices increase, so did the posterior probability of being an independent lineage. And so what were our research questions with these expectations in mind? First pass was, what's the concordance between the two? What's the proportion of coin and ore good species and MSC good species that we get out of these analyses? And then further going into more nuanced stuff, do we see that the MSC provides evidence for some of their conclusions about speciation theory regarding reinforcement and Haldane's rule. So briefly, very briefly, the methods that we use is we constructed a comparative data set by pulling sequences off GenBank using the sister pairs that coin and ore used. We pulled sequences from a suite of 25 genes. This was all, we read through the literature and determined which genes we wanted to use, and then we searched GenBank for these 69 species and these 25 genes. All told, we pulled about 2,600 sequences off GenBank. We then filtered that down depending on whether there was pre- and post-zygotic isolation measures, etc. And then we performed the MSC analysis species delimitation using BPP. The model of delimitation we used was the A11, which is just the joint estimation of species limitation and species street inference. And we used empirically derived priors for divergence time and population size estimates based on the literature search. So what did we find? First pass, well, we find that the MSC recognizes more independent lineages than Coin and Orr. And this is not surprising, right? Because they built their whole model based on reproductive isolation, and we're just looking for independent lineages. So the MSC, BPP in particular, comes up with 67 independent lineages, whereas Coin and Orr's definition comes up with 49 independent lineages, but together the two of them come up with 44 out of 87. So, I mean, yeah, BPP is doing what we thought it would. It's detecting the structure in the populations. These results, we use this posterior prior probability of 0.95 as a good species. I'm going off like recent literature that's been published on the debate between the two. We lowered this threshold to 0.90, 0.85, and the results changed a little bit quantitatively, but qualitatively it was the same result. So then we dug more into these nuanced differences, the reinforcement and Haldane's rule stuff. And we see that when you're looking at prezygotic isolation, they came up with this index that's done mostly in the lab. You get no choice, female choice, male choice, multiple choice, and then there's an index of prezygotic isolation. Ranges from negative infinity, which is complete disassorted mating, to 0 to 1, which is complete prezygotic isolation. So if we look at these in their allopatric and sympatric species, we see that in allopatry, we see that as soon as you cross a threshold of genetic distance, your posterior probability shoots up. The dots here are colored according to the posterior probability of being independent. So we kind of turn this on its head a little bit. We get posterior probability on the y-axis. These are all the same data points, but just colored differently. We have prezygotic isolation. They're colored according to prezygotic isolation now. We see that it's kind of all over the map. And this is kind of what you'd expect, because in allopatry, you don't really need prezygotic isolation to stop from mating with heterospecifics, because there aren't heterospecifics around. More importantly, to the Coin and Orr data set, they said in sympatry you see this evidence of reinforcement, and we also see it because here with low genetic distances, we have high posterior probability and with high prezygotic isolation, suggesting that, reinforcement is maintaining these species boundaries even when the genetic distance is very low. Again, we're going to turn it on its head the same way as before. And this is more clear. We see most of our high posterior probabilities are in that top left corner, where there's low genetic distance, high posterior probability, and high prezygotic isolation, suggesting reinforcement is playing a role in maintaining these boundaries in sympatric taxa. The other thing that we were interested in seeing if we could find support for was Haldane's rule, and this came out of their post-zygotic isolation studies. So there, again, they had an index that if you did these reciprocal crosses, you got a score of 1 if you were either sterile or infertile. So for example, for Haldane's rule, you would have to have the males, the heterogametic sex in this case, score 1, and then 0.5 is Haldane's rule under their scoring. So what do we see? So we have post-zygotic isolation on the x-axis and posterior probability on the y. And we see that as soon as you cross this threshold, as soon as you have post-zygotic isolation greater than 0.5, saying once Haldane's rule has been met, posterior probability of being independent lineages shoots up. And before we talked about whether it's allopatry or sympatry, and we thought maybe that would be playing a role, but it doesn't. It doesn't matter if you're in sympatry or allopatry. As soon as you cross this threshold of 0.50 post-zygotic isolation, your posterior probability of being an independent lineage shoots up. This is a pretty remarkable finding. This has nothing to do with the BSC or coordinates. I mean, we found this using the MSC, and there's support for one of the foundational theories in speciation as far as we know. So wrapping things up here, our findings from the MSC delimitation, they were in line with a lineage-based concept of what a species is. We found more using PPP than Coin did, but that's not really surprising. That means that both of them did what they said they were going to do. So we did find support for both reinforcement and Haldane's rule. This is pretty remarkable. As for reinforcement, we say that it's supported because there's high posterior probability, even for these recently diverged taxa that are in sympatry. And as for Haldane's rule, we see this pattern that if you have post-zygotic isolation greater than 0.5, that's a really strong indicator of a lineage split. And moving forward, we'd say evaluating these species delimitation methods based on an empirical data set is critical. Using an absolute model for, such as the Coin data set or any empirical model to do model assessment and accuracy and adequacy of your model, say the multi-species coalescent, is critically important moving forward. And ultimately, inference depends on having a good model. So if we don't test the model empirically using our understanding of the biological basis for species, the model is not as strong as it would be otherwise unless you test it empirically. So funding for this came from NSF and University of Hawaii. And with that, I'll take any questions. Okay, nice.